Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everyone. Welcome to lecture 6, Organic Chemistry 3. Today we will continue on amino acids, peptides and proteins. Today we continue on peptide structure determination. In the previous lecture, we talked about the determination of the peptide sequence using the N-terminal analysis. Well, today we will talk about the C-terminal residue analysis. Actually, there is no efficient method for sequencing several amino acids of a peptide starting from the C-terminus. In many cases, however, the C-terminal amino acid can be identified using enzyme carboxypeptidase. Well, in this case, we have the peptide incubated with the carboxypeptidase and monitor the relieving amino acid out of this reaction. So we get this part out. So using chromatography, as we build amino acid, which one comes first, this will be the terminal, and we get a new peptide residue. Again, the carboxypeptidase will react on this terminal, and then we will see this coming out next in the chromatography, etc. So the products are the free C-terminal amino acid and shortened peptide. Further reaction cleave the second amino acid that has now become the new C-terminus of the shortened peptide. Eventually, the entire peptide is hydrolyzed to its individual amino acid. Well, the Edman procedure using N-terminal peptide analysis is the most widely and successful method, but it is limited to a polypeptide with around 30 amino acids. For longer ones, it is necessary to cleave the larger chain into shorter fragments in a selective and predictable manner. This cleavage method relies mostly on hydrolytic enzymes such as trypsin, which hydrolyze lysine end unit and arginine from the carboxy end. Clostridione from its hydrolyzed arginine again from the carboxy end. And here thermolysine, leucine, isoleucine, valine from the amino end. Well, here is an example of selective hydrolysis of B-chain insulin by trypsin. Here we have arginine and we have lysine, and remember that trypsin will hydrolyze lysine and arginine at the carboxy end. So we cut in here and we cut in here, and we end up with three pieces. One fragment, arginine, will be the C end, and another fragment where lysine will be again the C terminal, and alanine. Well, here is another example. Show where trypsin and chymotrypsin would cleave the following peptide. Here is a peptide sequence with an amide end. Trypsin, as we mentioned, hydrolyze the peptide at lysine and arginine carboxy end. Chymotrypsin will hydrolyze the peptide as phenylalanine, tryptophan, and tyrosine carboxy end. Using trypsin, well, we have here arginine. We have here lysine and another lysine. So the peptide will be hydrolyzed at arginine carboxy end, lysine carboxy end, and here at lysine carboxy end. Using chymotrypsin, which again will hydrolyze the peptide at phenylalanine tryptophan tyrosine carboxy end. Here we have tryptophan, we have phenylalanine, we have tryptophan, and we have phenylalanine. Well, it will hydrolyze the enzyme or the peptide here at these ends, carboxy ends. Okay? A polypeptide containing 21 amino acids was hydrolyzed by thermolysin. The products of this treatment were 6 products summarizing here. And remember that thermolysin hydrolyzes the peptide at amino ends. So these are amino ends. Well, and when the same polypeptide was hydrolyzed by chymotrypsin, the products were 3 products here. Okay, remember again, the chymotrypsin hydrolyzed the peptide at phenylalanine tryptophan tyrosine at the carboxy end. So these are the carboxy ends. Well, how to solve this puzzle? We select the largest peptide sequence, which is G. Okay. Now, when we look for the first series, we find here glycine isoleucine. Okay, and we have again this part and this part are there, and leucine, tyrosine, glutamine, again they are overlap peptides. So we can connect these together, the series now from G, by adding or verify where E and F and A and D and C, we link C to G and we form another unit. Again, from the second chymotrypsin hydrolysis, we get this peptide. Remember that the tyrosine here is a carboxy end, so this will be the amino. Okay, here it is. We link these together and we create another peptide sequence. From the first group of hydrolysis, trypsin, we get the leucine, which should be an amino end. Okay, here it is. And we end up with this unit, which confirmed that structure is correct because this showed up in the second hydrolysis using chymotrypsin. So this is the acid end, carboxy end. So this is the new with all fragment from A to I. And yes, this is a 21 amino acid sequence. Peptide sequencing by mass spectrometry. A typical peptide can be sequenced in the mass spectrometer using electrospray or matrix-assisted laser desorption ionization techniques. The peptide in the gas phase is protonated to M+1 cozy molecular ion. In a special type of spectrometer, this ion can be selected and subjected to noble gas such as argon or xenon with high energy and high speed, so this ion will get fragmented. From the fragment and computer assisted programs, we can actually see the slices of the amino acid or amino acid unit. Compounds can have the same empirical formula, the same molecular weight, or M/Z, but different chemically. So breaking them into parts, what we call it fragmenting them, helps to identify what they are. Each of the following peptides rise exactly the same cozy molecular ion M+2, two positive charge ion. 1, 2, 3, 4. You can see they are single abbreviated peptide. Okay, so that's VFA, here is VAF. It's just the sequence is different. Okay, and what we need actually in proteomics is to distinguish between these peptides. For example, here we have tetrapeptide, that is the cozy molecular ion, and the collision of this ion with gas molecules like argon causes it to gain energy and undergo fragmentation. The fragmentation can be A, B, C, or X, Y, Z. What do you mean? A, we will see this part. B, we will see this part. And C, we will see this part. As M/Z, I mean the charged species. The charged species in X, Y, Z, for X, we will see this part charges. M/Z from this one. Y, we will see that part. Z, we will see that part. In many cases, B type and Y type fragmentation occur most frequently. That is fragmentation from the peptide or amide bond. Well, for A and B and C, we will see, for example, A2, this part, the ion will be double bond between this carbon and this nitrogen. It will be double bond, positive charge in here. Once it has a positive charge, then it will be seen in the mass spectrometer. B, well, for B2 we'll see that part. As a cesium ion. Okay? For C2, we will see that part as NH3 plus, the charge on the nitrogen. For X2, Well, for X, you will see that part. And the structure of ion will be acylium from the terminal. Okay? And here is Y2. What is Y2 here? So it will end up with nitrogen with positive charge on it. Z2, we will see that part, and the positive charge will be on that carbon. Here it is. For more, you can read the Biomedical Mass Spectrometry paper, volume 11, page 601, simplified at its letter to editor in 1983. Well, the scientist put the unit of the mass unit for this part of the amino acid with the abbreviation AL means 71, M over Z, arginine 156, etc. And this only for NHCHR C double bond O unit, this part. Okay, of course, for the amino acid, we still need one H here and OH in here. Okay, this calculation is made by only this part of the amino acid. Okay, and then with computer programs, okay, assisted analysis, scientists can find out the sequence of the fragmentation. So how does mass spectrometry works in proteomics? As we mentioned, we have calculated and provided computer program with the residue mass of these amino acid units. The Bn, which is the fragment coming out from the N terminal, will be equal to residue mass plus 1. On the other hand, the part coming from the C terminal will be equal to residue mass H2O plus 1. So if we have this peptide, the fragment coming from the N terminal, like B3, which means that we have this fragment A, D, G, we will see for A 71, D 115, and for G 57. So we will see a fragment at M over Z 244. The fragment coming out from the C terminal EVR E 129, V 99, R 156. Then we add 18 for HOH and 1. So for this fragment, if you have it, then you should see M over Z at 403.21. For the B-type fragmentation pathway, here we have the protonated or vicose ion, where the protonation occurs on this oxygen. Here we have the N-terminal residues, and here the peptide C terminal residue, and we have here amide bond. Lone pair will move to form a double bond, and here will attack on this carbon, breaking the double bond to form a single bond, leading to the formation of this species. Transfer of this hydrogen from that nitrogen to that one, well, will form a cation on this nitrogen. Lone pair will go down and kick this part out as a neutral species, forming the protonated oxazolone species, which is charged and will be seen in the mass spec. This part will not be seen in the mass spec, it's neutral. Or the protonation will occur on the nitrogen, and once this cation formed, then this lone pair will come down to form the acylium ion, and this will be again neutral part, and M/Z will show up. Okay, now this comes from the B ion. This C single bond can be broken, and CO will get out, and the fragment will be M/Z in U1 daughter ion. Okay, as if we actually cut in here. Okay, but the neutral species comes the peptide residue and CO2 neutral unit, and the charge will be on that one. So this one can be seen in the mass spec. See you next lecture.